Imagine a medicine that doesn't just treat your symptoms, but actually goes in and fixes the problem at its source, right inside your DNA. Well, that is the incredible promise of gene therapy. So let's dive into this amazing new frontier of medicine and find out how scientists are literally learning to rewrite the code of life itself.
And, this isn't science fiction. This isn't some far-off dream. Right now, today, in labs that look just like this one, that future is being built. Scientists are hard at work developing treatments that can repair, replace, or even just delete the faulty genes that cause some of the most devastating illnesses out there.
Okay, so before we go any further, let's get on the same page with the lingo. The official kind of fancy name for all of this is Advanced Therapy Medicinal Products, or ATMPs for short. Basically, it's the big umbrella term for any medicine based on genes, tissues, or cells, all of them designed to attack the root cause of a disease, not just the symptoms.
So, how does this all work? Let's break down the basic idea behind fixing our genetic source code. It's actually surprisingly elegant when you get right down to it. This quote just nails it. For so many diseases, think hemophilia or certain types of cancer, the problem really starts with one single defective gene. So the whole principle of gene therapy is, hey, what if we just deliver a correct working copy of that gene into the body's cells? Simple concept, right? But man, making it happen in practice is incredibly complex.
Now, this is a really important distinction we need to make. The gene therapy that's happening today is exclusively somatic. What that means is it only affects the patient's own body cells. The changes stop with them. The other kind, germline therapy, would alter reproductive cells, sperm or eggs, and those changes would be passed down through generations. That is not allowed, for some pretty major ethical reasons. So the key takeaway here is we are focused on treating individuals, not re-engineering the entire human gene pool.
Okay, but how do you actually get a new gene into potentially billions of cells inside a living person? I mean, think about that. For that, you need a very specialized toolkit, a gene delivery system. And there are two main strategies for doing this. The first is called in vivo, which just means inside the body. That's where the therapy is injected or infused directly into the patient. The second strategy is ex vivo, or outside the body. And this is where it gets really interesting. Doctors actually take some of the patient's cells out, fix them in a lab, and then put them back in. This here shows that ex vivo process perfectly. First, you isolate the cells you want to target, let's say stem cells from bone marrow. Then, in the lab, you use a special tool to modify them. After that, you grow millions and millions of these newly corrected cells. And finally, you reinfuse them back into the patient, where they can get to work and do their job. So what is that special tool? Well, it's called a vector. And honestly, this is just brilliant science. Scientists have cleverly repurposed nature's own delivery expert, the virus. They take a virus, strip out all the bad stuff that makes you sick, and replace it with a good, therapeutic gene. The virus becomes a tiny, microscopic delivery truck.
But not all delivery trucks are the same. This table gives you a quick look at a few common viral vectors. Some, like adenovirus, can trigger a big immune response. Others, like retroviruses, actually stitch the new gene right into the host's DNA for a really long-lasting effect. But that comes with its own set of risks. Honestly, choosing the right vector for the right job is one of the biggest challenges in this whole field.
All right, all this theory is fascinating, but where is it actually making a difference? Let's talk about some of the incredible breakthroughs that are already changing people's lives.
This is just an amazing story. ADS-CID, you might know it as bubble boy disease, is this devastating disorder where kids are born with basically no immune system. But with a therapy called Strimvelis, doctors can take a patient's own stem cells, use that ex vivo process to fix the broken gene, and give them back a brand new, fully functioning immune system. It's not just a treatment, it's a cure.
And it's not just for inherited genetic disorders. CAR T therapy is completely revolutionizing how we treat cancer. What scientists do here is take a patient's own T cells, the soldiers of our immune system, and they genetically engineer them in a lab to become expert cancer cell hunters. You are literally turning the patient's own body into a highly specific, cancer-destroying weapon.
And the results? They can be absolutely stunning. For some patients with very aggressive blood cancers, people who had tried everything else and run out of options, CAR T therapies have led to complete remission rates as high as 90%. That's a game-changing number.
But, and this is a really big but,
With such incredible power comes some incredible risk. For all of its promise, the road to getting here has been paved with some really high-stakes hurdles and very serious setbacks.
This timeline really shows you what a roller coaster the field has been on. After some early success in 1990, the field was shaken to its core in 1999 by the tragic death of Jesse Gelsinger, an 18-year-old who had a massive, fatal immune reaction to the viral vector in his trial. Then, just a few years later, another trial successfully treated an immune disorder, but some of the patients later developed leukemia because the vector had accidentally switched on a cancer-causing gene.
So those stories from the past really highlight the huge challenges that scientists are still tackling today. Safety is number one, of course, preventing those dangerous immune responses and making sure we don't accidentally cause cancer. But beyond that, there are big hurdles in just making sure the treatments work every time, for the long haul. And the manufacturing, it's so complex for these personalized medicines. And then there's the astronomical cost.
And that cost is a massive barrier. A single treatment of that CAR T therapy, Kymriah, was projected to cost almost $500,000. So how do we make these life-saving cures accessible to everyone who needs them? That is a huge question society is still trying to figure out.
Now, given all these high stakes, you're probably wondering, who makes the rules for all this? Well, the good news is, these powerful therapies are not developed in a vacuum. They are subject to incredibly strict regulation.
There's a whole global network of expert organizations in charge of oversight. You've got the MHRA in the UK, the European Medicines Agency, which has a special committee just for this stuff, and of course, the FDA in the US. Their entire job is to make sure these therapies are both safe and effective before they ever get near a patient.
Yeah, the path from a cool idea in a lab to an actual treatment a patient can receive is long, and it is painstaking. Every single one of these therapies has to go through years of rigorous clinical trials to prove its quality, its safety, and of course that it actually works. This whole evidence-based process is there for one reason: to protect patients. So, this is where we are. We have a technology with the power to cure diseases that were once considered a death sentence. It's an absolutely incredible moment in the history of medicine. But as these therapies get more powerful and more common, they force us to ask some really profound questions. What are the ethical lines that we as a society are going to decide should never, ever be crossed?